Good morning, everybody. I am Hudu Singh. Today we are going to discussing about the application of paper chromatography. Firstly, we are discussing discovery of paper chromatography. Paper chromatography was employed by scientist Michael Twist in 1900. In 1952, A. Martin and R. Singe won the Nobel Prize in Chemistry for development of other chromatographic techniques. Then, paper chromatography. Paper chromatography, as the name implies, is carried out on paper. What is paper chromatography? How can we define it? Chromatography technique that uses paper sheets or strips as the absorbent being stationary phase through which a solution is made to pass is called paper chromatography. It is an inexpensive method of separating dissolved chemical substance by their different migration rates across the sheets of paper. It is a powerful analytical tool that uses very small quantities of material. Then, what actually happens in paper chromatography? As we all know, ink is a solution containing a number of different molecules, different characteristics of a as solubility and size are present in this molecule. When pulled along the piece of paper toweling, each molecule travels at a different speed because of their different characteristics. Compared to the heaviest particle, the lightest particle move more quickly and at the greatest distance. The pigments that make up the ink sample are thus separated out. Then we are going to our main topic, application of paper chromatography. Paper chromatography finds many applications in inorganic, organic, biochemical and industrial fields. Some of these applications are: first, a paper chromatography is particularly suitable for the separation and identification of sample components present in the trace quantities in microgram levels. For example, it can be applied to separate a mixture of dyes, Congo reds, phenol reds, and bromophenol blue in a solution by using butanol ethanol NH4OH mixture as a developing solvent. Then the second application, it finds application in the separation of components which are closely related chemically. For example, a mixture of amino acids like arginine, glutamic acid, leucine, and valine can be separated by the paper chromatography using butanol acetic acid water mixture as a developing solvent. Then the third application, it is applied for the control of purity of food products and the pharmaceuticals. For example, the different food colors used in the food sample can be separated and identified using paper chromatography using propanol as a eluent. And the fourth, it can be applied for the separation and identification of metallic cations present in a solution. Here the example, it can be used to separate the cations like Fe3 plus Ni2 plus and Cu2 plus in aqueous mixture of their salt using acetone hydrochloric acid mixture as an eluent. The individual cation can then be identified by spraying the paper with suitable spot test reagent or comparing with their RF values. Then the fifth, it is used in the laboratory and in industry for check of purity of organic chemicals. Pure commerce give one spot. And the sixth, it is used to dictate drugs and dopes in animals and humans. Seventh, it is used in the analysis of cosmetics. 8. Resolution of isomeric metal complexes can be achieved using paper chromatography. Also, it is used in the determination of metal in our sample. For example, the detection of aluminum and chromium in the metal ore can be done by the Hayes phenomenon. Then the 10th, with the help of this chromatography, we can study ripening and fermentation phenomena. 11. Detection and estimation of adulates and contaminants in the food material and in drinks can be done. And the 12. The phenomenon can be used for the analysis of corrosive reagent. And the 13th, it is useful in the separation of plant pigments. And the last, pathology and forensic science. For investigation of crimes, paper chromatography is useful in the fields of forensic science as this process can be successfully carried out with even very small quantities of material. Using this technique, samples from crime scenes are collected to be analyzed and identified used in DNA and RNA fingerprint. Moreover, to detect the presence of alcohol or chemicals in the blood, pathological laboratories use paper chromatography. Now, paper chromatography offers many advantages like low cost, unattended, hassle-free operation and simplicity. But there are some limitations too. First for limitation, the development of chromatogram requires long time, about 24 hours. So it is time consuming. So process is slow. Then it does not give accurate result for larger concentration. Sensitivity of the method is low. Now we are going to conclude this presentation. Ranging from forensic investigation, pharmaceuticals, environment monitoring, and foods, paper chromatography have retained their ground in various fields. For matching the requirements of high throughput laboratories, chromatography has seen phenomenal growth in terms of software applications, increased in automation and separation in general. So I hope you are little clear on applications of chromatography. Thank you so much for listening my words and seeing my presentation.